Hello everybody. My name is Michael Hamblin. I am presenting an invited talk on antimicrobial photodynamic inactivation with inorganic salts. For 25 years, I was at the Wellman Center, Mass General, Harvard Medical School, but then I retired and I'm now affiliated with the University of Johannesburg. So the outline we have here, we do aPDI because of multi-drug resistance, and then I'll talk about the different salts: azide, iodide, thiocyanate, selenoscienate, and nitrite. So the problem of antibiotic resistance is widespread, caused several reasons: unnecessary prescriptions, people stopping because they're cured, and even use in animal feedstuff. So widespread problem. And one way of overcoming it is with antimicrobial PDI, and here is the Jablonski diagram showing the photosensitizers getting excited to the singlet state, undergoing an intersystem crossing to the excited triplet state, and from thence they can undergo type 1 or type 2 photochemical pathways, forming singlet oxygen or hydroxyl radicals. And these are very reactive oxygen species that can attack all known biomolecules, amino acids, proteins, nucleic acids, fatty acids, and they can basically kill everything, including all kinds of pathogens. But mainly today we're going to talk about bacteria, a little bit about fungi. So the advantages of using aPDI as a treatment, i.e. aPDT, are it's safe for tissue, inexpensive, works rapidly. Patients don't have to remember to take pills, doesn't need training. You should remember that systemic antibiotics cannot get into dead tissue, and even if antibiotics work, they take several days or weeks. You don't need to know what the pathogen is precisely before you start, whereas with antibiotics you do. You can kill pathogens and biofilms, and PDT itself does not induce the development of resistance. With bacteria, you can have gram-positives and gram-negatives, which have different cell walls, so you need to be aware that certain photosensitizers can kill both kinds, but in order to kill gram-negatives, you need cationic photosensitizers. Whereas to kill fungi, again, you tend to need more lipophilic photosensitizers. So there is a bit of a dependence on structure for the appropriate photosensitizer to kill different kinds of bacteria. So we got into this field quite a long time ago now, 9 years ago, when we discovered by accident that sodium azide, rather than inhibiting the killing of microbial cells, bacteria, actually paradoxically potentiated them. We expected that we would quench singlet oxygen and inhibit killing, but when we used methylene blue, we found the opposite, that we could get more killing. So roughly about 2 logs of extra killing, so 100 times more effective when you put azide in. But when you used a photosensitizer that works exclusively by singlet oxygen, this PIC6 conjugate, where azide almost completely inhibited the killing. So methylene blue works by type 1 and type 2, and remember that type 1 is radicals, so the hypothesis was that we got azide radicals from type 1, an electron transfer, and these azide radicals could kill the bacteria. So this gave us an idea that we could make other radicals from inorganic salts, for instance, iodide radicals from potassium iodide. But amazingly enough, we found that as opposed to azide, all photosensitizers gave remarkable potentiation with potassium iodide. And we're talking here 6 logs of extra killing, like 1,000,000 times more killing. So this is not a small effect. This is photophyrin, which is well known not to kill gram-negative bacteria, but here you see it can kill 5 different gram-negative species, 6 logs of extra killing just by adding iodide. Interestingly, if you mix the photosensitizer and the light, in other words, you shone the light on the photosensitizer plus the potassium iodide and then added the cells afterwards, you still got 6 logs of killing if you had enough Ki in there. So with photophyrin potassium iodide, you're making something that will kill bacteria, and the obvious answer is free iodine because you're oxidizing iodide to iodine. Works with gram-positives. It also works with fungus, Candida albicans. When you get to 100 millimolar Ki, you get several logs of extra killing. 100 millimolar you might think is quite a lot when you're only using nanomolar photosensitizer or micromolar in some cases. But when you think that the potassium iodide has to react with a short-lived ROS, singlet oxygen, you need a high concentration to trap it. So here's the mechanism of action. First thing, you get addition of singlet oxygen to iodide to give you peroxyiodide, which can then undergo a sort of rearrangement and give you a mixture of hydrogen peroxide and free iodine, but you also can get iodine radicals. So you can either get radicals or you can get free iodine and hydrogen peroxide, which are synergistic together. With rose bengal, similar results. Rose bengal normally does not kill gram-negatives, but when you add Ki, you get a lot of extra logs of killing. Obviously, gram-positives and Candida, the same thing. Because rose bengal doesn't bind to the bacteria, if you spin the dye in the cells to remove any non-bound dye, it actually doesn't work particularly well with gram-negatives. Again, if you put the cells in after you've shone light on the dye and the potassium iodide, then it works pretty well. If you use an animal model with bioluminescent bacteria, you can go up to 20 joules per square centimeter. Nothing much happens unless you have potassium iodide in with the rose bengal in the mouse wound. And you can make a graph because you can measure these bioluminescent signal from the imaging system. We had 2 porphyrins, tetra-substituted porphyrins. One was tetrasulfonate, so it was anionic, and the other was a tetrapyridinium salt, so it was cationic. Turned out that when you put potassium iodide in, something quite interesting happened, that with E. coli, whether you'd spun the mixture of the cells in the dye or not, or put the cells in after, you got exactly the same result. Now, this was surprising because we did not expect TPPS4 to bind to the gram-negative cells because it's an anionic porphyrin. We've shown that this works, adding potassium iodide works in some more applicable situations. This was a study looking at E. faecalis, which in dentistry is a big problem in teeth and endodontic infection. And here you're looking at, again, methylene blue with red light, planktonic cells, biofilm cells here, transmission electron microscopy. So the combination of methylene blue and Ki has a much bigger effect than just the methylene blue PDT alone. This was a model in female rats with a bladder infection. So again, we used bioluminescent uropathogenic E. coli into the female rat bladder, and you can put a fiber through the urethra into the bladder, so you can illuminate from the inside. And again, you do methylene blue PDT alone, nothing much happens, but you put the potassium iodide in, and you can eliminate the bioluminescent signal. And here you see a follow-up for up to 5 days after the PDT treatment, single treatment on day 1. And you see that when you have KI, a lot of the rats, you do not have any recurrence. Occasionally you do, but mostly you don't. This is a clinical study in patients where we're using oral candidiasis. Well, patients have oral candidiasis, so pilot clinical trial, putting methylene blue into the mouth and adding potassium iodide onto the methylene blue stained lesion and then shining red light. And here you see if you do 2 successive treatments, I think they were 1 day apart, I believe, you get quite a good clinical effect after the second treatment. So potassium iodide worked well. Here we're looking at potassium bromide, but the only way to get any potentiation with potassium bromide is by using titanium dioxide photocatalysis, which is a much stronger way of generating ROS compared to most PDT. And here you see if you use potassium bromide, you can get, some logs more killing, not 6 logs more, but a couple of logs of extra killing by adding sodium bromide to antimicrobial photocatalysis. And the reason you need the bromide, as I said, is you get this large band gap semiconductor. Here you go, 3.2 electron volts, which is enough reducing power to reduce bromide to probably hypobromite. That's what we think the active species is with bromide. Then we went on to look at thiocyanate. Again, it's a simple inorganic salt, non-toxic. But we found that, interestingly, we got sulfur trioxide radical anion, and the mechanism involves single oxygen. So here you see several logs more killing when you increase the potassium thiocyanate concentration, both with Staph aureus, E. coli, in a light dose dependent manner. This is a spin trap showing that we get sulfur trioxide radical anion. And this is what we think the mechanism is, that you're adding singlet oxygen to thiocyanate to get an intermediate, which decomposes to sulfur trioxide, and then you get the radical anion. And this is the species that's giving the extra killing of the bacteria. So thiocyanate worked. What about selenoscyanate? And selenoscyanate turned out to be even better than thiocyanate, remarkably better, actually. But here the mechanism turned out to be different. We think that we're making selenocyanogen, SCN2. So here you see with the selenoscyanate concentration, you get extra killing both with methylene blue, res bengal, and the tetraanionic porphyrin, and also with Staph aureus. When you put the cells in after the light, you get killing, so you are producing a toxic product which at least has some stability. Here you see that gram-positives and gram-negatives, you get killing when you put the cells in after light. And if you put them at different times after the light, you see that the cytotoxic species you've generated slowly decays, and it decays over about 20 minutes. So whatever you're making that's killing the cells has some stability, but not infinite stability. Works for a lot of different gram-negative species, a lot of different gram-positive species, regardless of whether the cells are in there or you add them after, you get the same umpteen logs of extra killing, 5 or 6 logs of extra killing. It works the same with lead tetraacetate. So lead tetraacetate on its own doesn't really kill bacteria, but when you put the thiocyanate in, it kills 6 logs. And the interesting thing is that lead tetraacetate is a literature preparation of selenocyanogen. So the fact that this worked suggests that where the single oxygen is reacting with the selenoscyanate to form selenocyanogen. We thought we'd compare thiocyanate and selenoscyanate to see if we can draw some comparisons between the two. Obviously, selenoscyanate is a lot better. The only thing that works with thiocyanate is methylene blue. So rose bengal and the porphyrin don't really work at all here. Whereas selenoscyanate works with virtually all of them and works a lot better. Thiocyanate doesn't really work to any extent when you put the cells in after the light. So you're not making a stable antimicrobial species with thiocyanate, but you are with selenoscyanate, as we saw here. Fenton reagent, which is ferrous iron and hydrogen peroxide. If you add thiocyanate and selenoscyanate, you get a little bit of potentiation of killing. Both with E. coli and with MRSA, it's more pronounced with selenoscyanate than with thiocyanate. Horseradish peroxidase, another way of generating reactive oxygen species. Selenoscyanate gives you a lot of potentiation compared to thiocyanate, and with MRSA especially. So the selenoscyanate is easier oxidized and gives more bacterial killing. Again, comparing lead acetate, it works a bit with both of them, but it's more pronounced with selenoscyanate. So the proposed mechanisms, thiocyanate, sulfur trioxide radical anion, but only with phenothiazinium dyes. With selenoscyanate, we think we get selenocyanogen, any photosensitizer that produces singlet oxygen. Lead tetraacetate is a literature prep for both thiocyanogen and selenocyanogen. Horseradish peroxidase, in the literature, it is reported to produce hypothiocyanate, but hyposelenocyanate is said not to exist. So it's unclear what the mechanism is here. And with Fenton reagent, there's not really anything in the literature precisely to suggest what's going on. So finally, to look at sodium nitrite, again, a simple, harmless salt, and again, it can potentiate aPDI. Can't really see, it's a bit small to see these graphs, but when you add the nitrite and the light, you get extra killing, in some cases a lot extra killing. And what we think is going on, we did show that If you did this in solution with no bacterial cells, you could nitrate tyrosine. If you had the photosensitizer, nitrite, salt, and light. And if you can nitrate tyrosine, you're producing a reactive nitrogen species for sure. And after a bit of investigation, we came to the conclusion our favorite intermediate would be peroxynitrate, which is what you get if you add singlet oxygen to nitrite. You get peroxynitrate. Most people would say, well, it's peroxynitrite, but no, because these reactions with singlet oxygen are addition reactions, we hypothesize that it is peroxynitrate. So the conclusions, the photochemical potentiation involves adding a relatively simple chemical compound, non-toxic salts, has complex mechanisms. Many salts work: iodide, bromide, thiocyanate, selenoscyanate, nitrite, and even azide. But since azide's toxic, this one would not have clinical applications. All these salts, and even selenoscyanate, are relatively non-toxic. Electron transfer reactions from excited phenothiazinium dyes appear to be important. Titanium photocatalysis is the only one that's potentiated by bromide. Iodide is clinically relevant. It's already approved for use in patients, and we did a preliminary clinical trial to show it works in oral candidiasis and AIDS patients. I didn't show you the data today, but oxygen-independent photoinactivation may be possible using some of these salts. So the acknowledgements, a lot of people have worked on this project over the 9 years we've been doing it. Tad Sana from Poland has done a lot of the mechanistic work, a lot of other folks in the lab, and various funding agencies. So I will wrap up there. Thank you all for listening, and let me stop it here.